Hello, my name is Sean Kimbro from North Carolina Central University in Durham, North Carolina. And I want to first thank the organizers for inviting me to share with you some of my lab's efforts to solve or at least address or understand cancer health disparities. The one aspect that you must appreciate when you think about cancer health disparities is understand that what the problem really is, or at least understand what the problem looks like within the parameters of your investigation. And so I tell my students a lot of times that understand exactly what you are aiming to resolve or to solve, and the so what behind that research or that bench experiment. And so the American Association for Cancer Research actually gave a nice report just recently showing and describing the cancer disparities issue with regards to prostate cancer and breast cancer as you compare African American men and women respectively to their white counterparts. And you can see clearly there that you're talking about 111% increase in risk of dying from prostate cancer in African American men and 39% higher risk of dying in Black women when it comes to breast cancer. What's also notable here is that you scroll down, there are various other cancers, and a lot of them have a tendency to have ethnicity tied to them, or some type of association with a characteristic or a phenotype or a category. And so here, when you think about that, you need to ask, what are all the pieces that make up or that lead to that cancer disparities? And so I always like to show this idea that some of the key determinants of health disparities are host, the environment, and the behavior of individuals. And I always, as a bench scientist, concentrate and look at that genotype. And I like to start there because of my training. Over the years, I've appreciated this idea of environment and behavior, and of course, quality of healthcare and access to healthcare as being another major contributor to the disparities we see in mortality related to prostate and breast cancer disparities. So for the purpose of this talk, we're going to kind of concentrate on the bottom half here, starting off with the host and understanding the genotype and the impact of environment with that genotype and behavior with that genotype in a little sprinkle of the environment into that host and behavior mesh as it pertains to risk for prostate or breast cancer. Now, when we started this a while back, we didn't think, back in 2010, 2008, we didn't think that it would really lead to this level of response or interest. We realized early from some preliminary work looking at minor allele frequencies between populations that some of the variations between African American and European American, when we looked at minor allele frequencies, turned out to be in their immune-related genes, or those that were involved in immune regulation. And we realized that, wow, maybe that's a difference in order to explain for some of the large differences we're seeing in risk and outcomes. So even more recently, it's been suggested that genetic predisposition in immune modifiers in African Americans might contribute to poor prognosis. And even more recent, this idea of African geographic ancestry and the evolutionary selection of a more robust immune response or a different type of immune response related to ancestry might really play a role in the tumor microenvironment, especially in breast cancer and disease progression. Well, to appreciate that, you have to understand and respect the fact that life started in Africa, and out of Africa, human beings migrated out into the Americas. And in doing so, you have to step back and ask yourself that the most diverse population, the most diverse genotype, is actually in those of African descent or those that are from West Africa. And you see some of the newest genomes are actually in the Americas and Europe. So with that, you have to also argue that whatever the human beings were optimized for genetically in their environment in West Africa, that optimization would probably be a little different than what you would find in Europe and in the Americas. We actually addressed this issue in a publication in Cancer Immunological Research 2019, where we were looking at and proposed this idea that your ancestral environment, in this case, we were looking at breast and prostate cancer disparities in those of African descent. We realized that the selective pressures in that environment would have optimized your immune system such that you would have been optimized for that environment. And when you are forced through forced migration through slavery and the slave trade, and you are now transported from your ancestral environment to your non-ancestral environment here in the Americas, then you must ask yourself, wow, there's no time for change. Some would argue that there was a forced selection via the trip from West Africa to the Americas. But during that time, you would not have received necessarily any, or you wouldn't have gotten any selective pressure on the genome. But there's this migration from one environment to another, and then that change in that new environment, the different pressure in that environment, and not having enough time to adjust to those changes, if you had walked the land bridge, leads you to kind of appreciate the possibility that you would have a different innate immune response or a different immune response with the different pathogens, and that might increase the rate of cancer incidence or aggressiveness, and would honestly lead to possibly cancer disparities. So it's this framework that we've kind of worked with moving forward early on in trying to explain some of the differences that we see with regards to the biology of health disparities, of cancer disparities. So just to give you a little idea, this is a real short version of a little truncated version of the innate immune system. We're talking about toll-like receptors, and we're talking about the first response, simply put, in the immune response. Pathogen is recognized by a toll-like receptor. Those receptors then transmit a signal down through MYD88, IRAK4, or through TRAM and TRIF in terms of TLR3. And that communication downstream adapters and kinases leads to inflammatory proteins being released, cytokines, and also interferons in the case of TLR3 and 7/9. Also, you could go down the path and also, of course, end up at apoptosis. So, with appreciation of the pathway, you can argue. I'm going to present today TLR3 as a point of interest. IRF3, which is an interferon responsive factor. We'll look at IRAK4, and we'll see a little bit of evidence related to TOLIP and how that, and the variants that might occur in that might impact incidence in disease progression, or at least risk in terms of African American men. So the first project that kind of started this was a very small study of African American women at Grady Hospital in Atlanta, Georgia, while I was at Emory University in conjunction with some of my colleagues at Fox Chase and University of Louisville, Dr. Lucrece Kidd. And there, we identified some interesting variants that looked like we had some promise. There was a variant in TLR3 that actually was protective, and there was a variant in IRAK4, which was relevant in terms of possible increase in risk, up to almost 5 times greater risk. However, that wasn't significant. So the one thing that we've done, we realized that from that study, that was the first time we kind of recognized this TLR3 genotype, or TLR3 or a toll-like receptor being relevant in some breast cancer risk among an African American population. And that actually was associated with an 82% decrease in breast cancer risk. The IRAK4 story, we followed for a fair amount of time. It was only significant with risk under an additive genetic model and a P per trend, which was significant. Well, that was the beginning of appreciating at least the possible role of a TLR, or the toll-like receptor pathway, in the development of risk for breast cancer or cancers related differentially, disparate between African Americans and European Americans. We went a little further next time in collaboration with Camille Reagan at Fox Chase. The team worked together, and we wanted to do something a little different. Apply the genome, the host, the genotype, with the environment, and do it in such a way just to compare 2 African American populations that were not in the same location. And with the hopes that maybe that would get at a little bit at this idea that it's not just the genotype. We appreciate the environment as being a key component of cumulative risk when it comes to disparities in the mortality due to breast cancer or prostate cancer. And in this case, looking at men of African descent, when we looked at our gene list, our SNP list, and that SNP list, to let you know, actually came from the observation that some of the SNPs that we were interested in were not on the standard GWAS at the time, the GWAS chips at the time. And we made an effort that we would select those variants that were 25% differential between African American descent and Caucasian or European. And when you talk about the 1,000 genomes, it was the African genotype versus the actual European American group from Utah in that 1,000 genomes data set. And when we did that, we realized that we had a unique list, and this list was not on the chips. And we took that list and we started to apply it to African American samples. And like I said before, that was the breast cancer sample from Grady Hospital. And here we're presenting data from men mostly from Washington, D.C. area and men from Jamaica. And so what we found was that TLR6, toll-like receptor 6, also had an increased risk. If you had the variant, 1.4 times as higher. And then you had TOLA, which was protective, depending on which SNP you were looking at. It was protective if it was the 3' UTR variant, or intron 1, it was up to 1.5 times more increase in risk. So we wanted to do something a little bit different, though. Camille and Lacrease Kidd and Camille Reagan actually had a fantastic idea, and they separated out the U.S. men from the Jamaican men. And in doing so, some observations were made. We found out that one variant in the U.S. men was significant, but it wasn't in another. And here's a case where you had in the age-adjusted odds ratio in U.S. men was actually protective, where in the Jamaican men it was not. was not. And that was significant. What was also interesting that, and that was for IRAC4. And then we went to look at TLR6. Remember previously it did increase risk. However, the increase in risk really was a result from the Jamaican men, in which the Jamaican men had a twofold increase in risk with this variant. And we didn't see that in those US men. So that kind of brings into the picture this idea that location made a difference, even though they were from the same or similar ancestry. Further analysis was done using multi-dimensional regression, machine learning method to kind of create or identify rather relationships and associations. And by using MDR, we were able to identify a synergistic relationship with IRAC4 and TLR2, implying that together they probably have an impact on risk. Our numbers were not high enough to reach a threshold to really confirm that. But we did identify, of course, another set that was redundant. So the green is redundancy and red is synergistic and orange is in between. So at the end of the day, you can use this technique to look at relationships between IRAC4 and TLR2. Modifications to MDR exist and are available in the literature as well. What is interesting that this relationship that we see here is only found in prostate cancer risk for US men of African descent, not in the Jamaican men. So at the end of the day, we would argue that looks like we found a relationship between IRAC4 and TLR2 as possible prostate cancer markers for African American men of US citizenship or US from the US or in the US. However, that did not hold up for those men from Jamaican descent or those men that are in Jamaica. We would argue, this is when the beginning of our arguments, that the TLR-related genes, these toll-like receptor genes, might actually be responsible or impact cell survival, proliferation, and or inflammation. So it gets you to another piece. Now that we've talked about the environment, how about that behavior? And so when we start looking at that behavior, we're talking about diet, we're talking about exercise. I'm just going to show you one slide from a paper done in conjunction with using the same SNP list that we created way back and asked the question, is there a relationship between these SNPs that are reflective of a minor allele frequency difference between African Americans and European Americans? Can you use that SNP list to evaluate the risk as it relates to waist to hip ratio, for example? And this population here is 100% Jamaican. So this is not the US population here we're looking at. And what you can see clearly in the top half, you see the SNPs and the associated genes and their risk as it relates to being non-obese, to being lean. And below in the red box, you see that there's an increase in risk from 3 to almost 5-fold increase in risk if you have a WHR, a waist to hip ratio greater than 0.9. And these are significant relationships and associations. So you have to ask yourself now, we now have taken ancestry or a variation that is related to ancestry, and we now added a behavior and put those two together and shown an increase in risk or a risk associated with a certain genotype. And so obviously these associations are associated with obesity. They're suggestive. There's a small population, a small study relatively, but it definitely warrants looking further ahead in a population of men that are non-obese and obese of African descent, whether it be in the United States or across the globe. Another aspect recently published from Kamel's group in collaboration with Kamel Reagan from Fox Chase is IR3. And the IR3, remember, is in that pathway downstream from the TLRs that are associated with viral detection and response. And in doing so, we find that IR3 is protective and is significantly associated with being the protection of related to prostate risk overall in that population. But what's really interesting is that when you come and you add the history of their sexually transmitted infections, you find that it holds up even more. That there is a correlation between a variation in IR3 in the viral pathway that is correlated to the genotype and to their STI infections. And so we get at this story that at the end of the day, that we now have behavior, environment, and a genotype related to ancestry and related in part to environment where the individuals live or have lived for a couple of generations. And you find that you have a relationship that is truly implying a relocation or a response possibly to a novel environment, an environment that they were not optimized for originally. So it always leads to this slide where we talk about geographical ancestry as the original environment. And then that environment, one would have had to manage energy differently and the pathogen insult according to that environment. So if you came from West Africa, you can imagine you have heat, you have sunlight, and you have a pathogen component to this selection process, to the innate immune, and actually the whole individual, the whole genotype, that over time would have been modified. And that modification would be seen in such things as the innate immune genes, some transporter genes, and you would have such modulation of the lipid metabolism, glucose metabolism, that would be kind of unique to that population because of the environment. If you take them out of that environment and move them to this new environment, now you have this increase of cardiovascular metabolic diseases, things like asthma. You have things like triple negative breast cancer and prostate cancer. These new problems per se that might be a result more of the taking someone out of context. You're taking someone out of their original genetic or genomic or genotype context. And so I always like to leave something for people to think about as we talk about health disparities and genetics and the biology of health disparities and cancer disparities in particular, is keep in mind this idea of context. If you take an individual out of a context where they were optimized to survive and live, and you take them and you immediately impose them into another environment with different behaviors and different political systems and different social structures, it's kind of hard to think about how you translate some of that through xenografts, mouse models, or even cell lines. The idea of taking a variant and you take that variant and you identify it and then you put it into, let's say you identify a variant from an African-American population, you put that into a European-American context in terms of a cell line. And now you got a really different question because that variant would be out of context. And the same thing with the human beings, you've taken them out of context and not given them enough time to adapt to this new environment. And with the change in society, that the political and the social changes that have come along with time, you now have a change of behaviors that are a result not only of the environment, but also the political environment and some other parameters that are related more to the social and social behavior, including stress and so forth, that would have an impact on that genotype and thus impact the disparities. Another example of something to think about out of context or the implications of ancestry in conjunction with Dr. Kittle's laboratory at City of Hope, genetic ancestry that led to the misclassification of a cell line designated as African American for years, also makes you think about and keep in mind that the biology of health disparities is just as important, or at least is a player in the psychosocial and the behavioral piece, as well as the political ramifications to health disparities. And so I want to leave you there with that thought, with some of those things to think about. I need to acknowledge my collaborators at Central, my students and my postdocs in the laboratory, Susan and Erica, my friend at Fox Chase, Camilla Reagan, Lacrease Kidd, also Shanita at Jefferson, and our colleagues at the University of the West Indies. And I want to acknowledge the African Caribbean Cancer Consortium for providing access and sharing, being part of a team. It's been really, really nice to be part of a team that has a central mission at the core. I also want to thank the funding agencies, NIMHD, the National Institute of Minority Health and Health Disparities, and the National Cancer Institute for their support of this study, of these studies over time. And so I want to once again thank the administrators and the organizers for inviting me to speak today. And I look forward to any questions or any comments as we move on. Thank you very much. I hope it was informative. Thank you very much.